Hello, welcome to the Geriatric Nutrition course. My name is Dr. Abhirup Das. Today I am going to talk about an important molecule called NAD+ or nicotinamide adenine dinucleotide. It plays an important role in our metabolism, and recently it has received a lot of attention for its role in aging. But before we talk about its role in human health, let's walk you through the history of NAD+. A century back, the role of NAD+ was uncovered for its involvement in fermentation reaction, a reaction by which yeast converts sugar into alcohol, and that's how your beer is made. For this work, Chelpin from Sweden and Arthur Harden from London were awarded Nobel Prize in 1929. Its link with human health became prominent with this disease called pellagra. Pellagra is a nutritional disease that causes NAD deficiency in your body. It was a big problem in Europe long time ago. Some of the clinical features of the disease are dermatitis, diarrhea, and dementia. It was also called the disease of 4 Ds, and the 4th D was added for death because the disease was very fatal. In 1735, Kesel first described this disease in Spain, where people were living with poverty, they were malnourished, and they were eating mainly corn diet, which was introduced to them from Central America. In 1897, it became a big problem in South Africa when rinderpest outbreak happened. It's a kind of cattle plague that killed a lot of their cattle, and people shifted from milk and meat-based nutritious diet to maize-based diet. And on that time, America was recovering from Civil War, and pellagra was a big problem in southern part of United States. So United States government commissioned Goldberger to find a cure for pellagra. He was the first one to find a link between diet and tryptophan deficiency with this disease, and he did a lot of human experiments. Why tryptophan? Apparently our body uses tryptophan, which is an essential amino acids, by this pathway called de novo synthesis to make NAD+ in 8 steps. And in this process it uses ATP, which is an energy molecule, and amino acid glutamine. So before Goldberger can find a cure for pellagra, he was decommissioned because his experiments become very controversial. He started using prisoners and started giving them pellagra by withdrawing some nutrition from their diet. Thankfully, Elvejam from Wisconsin-Madison jumped in and used dog as a model and discovered nicotinic acid or niacin as a cure for pellagra. He called it pellagra preventative factor. So niacin is another molecule that also forms NAD+ in our body. It uses a different pathway called Price-Handler pathway. And this pathway was discovered by Jack Price and Philip Handler in 1958 at Duke University. So you might be wondering, what has to corn do with pellagra? And it's just not that corn diet that causes pellagra. It's a combination of malnutrition and corn diet. Apparently, when you don't get tryptophan and niacin from other diet, you don't get that from corn either, because the corn proteins are low in tryptophan. Moreover, the niacin that is present in corn is present as nicotinyl ester that our body cannot break down. So you can see how the deficiency of tryptophan and niacin not coming from diet can cause NAD deficiency. Now if that scares you from having a good nice Mexican food or tortilla tonight, don't be scared, because pellagra was not that common, or it was never a problem in Central America, which is the home of corn and where the whole world was introduced to corn. Why? Because apparently the way they make the tortillas is very different from the rest of the world. Instead of grinding or milling the grain, they actually soak the corn in lime water overnight and make a dough out of it. And during that time, lime water, which is alkaline, hydrolyzes the nicotinyl ester and free up the niacin, which our body can absorb. So you see, not the nutrition, but how you cook your food is very important, how you actually make the nutrition available to our body. Now, are these only 2 pathways by which our body can make NAD+? Apparently, NAD+ is used by different enzymes in a very quick manner and needs a repeat turnover, quick turnover. So our body can just rely on these 2 pathways that takes 8 steps and 3 steps to make NAD+, because you need to make it very quickly. So the 3rd pathway that is predominantly used in mammalian cells and in our body is called salvage pathway. In this pathway, our body uses nicotinamide, which is a byproduct of NAD+, to make NAD+. So here nicotinamide is actually converted to nicotinamide mononucleotide, which is a molecule that I worked on in my lab as well. and using the enzyme called NMNAT, it's converted to NAD+. So this connection between nicotinamide mononucleotide, NMNAT to NAD+ was actually discovered by another Nobel laureate, Arthur Kornberg from Stanford University. He got Nobel Prize for something else, but he's the first one to show that if you take NMN and NMNAT, you can actually make NAD+ in a test tube. So not only nicotinamide, but there's another molecule, another NAD precursor called nicotinamide riboside that can also make NAD+. It also uses the same salvage pathway. And it was a big discovery because this is something that you can get from food. And this was discovered by Charles Brenner in 2004, where he showed that this NR is highly prevalent in cow's milk. So how is NAD used in metabolic reactions? So one of the classic role of NAD+ is to be used as a cofactor in redox reactions. So some of the redox reactions you might be familiar with from your first year biochemistry. Some of them are, for example, glycolysis, by which our body breaks down glucose, and in this process NAD+ is used and converted to NADH. Another pathway is fatty acid beta oxidation, by which our body burns fat. It also uses NAD+. And the third one is called Krebs cycle or TCA cycle, which takes place in the mitochondria. In this process, NAD+ is reduced to NADH, and this NADH is shuttled to mitochondrial complex 1, which uses it to make ATP. And in that process, NADH is actually oxidized to NAD+, which keeps recycling. So this reaction is very important, in which NAD+ is reduced to NADH using the hydride transfer reaction. And this was discovered by a famous scientist called Otto Warburg. You might know him from his work on tumor metabolism and what we now know as Warburg effect. So what are the other ways NAD+ is used? So one of the most important role of NAD+ is to be used as a substrate in different reactions. And NAD+ can actually promote DNA repair by being used this way. So this was discovered by Mendel and his colleagues in 1963. Until then, most of the scientists focused on how NAD+ can be made or how NAD+ can be used in redox reactions. And this was a big breakthrough around that time, because Mendel was the first one to show that NAD can be broken down into two parts using this enzyme called PARP. PARP stands for poly-ADP ribose polymerases. It is an enzyme that uses NAD and takes the ADP ribosyl group, which is here I am showing, and adds this group to the lysine, glutamate or aspartate ends of proteins. And it can add up to 200 ADP ribosyl subunits to a protein. And in the process, it releases a nicotinamide, which can recycle back to NAD+ by salvage pathway. Why does it do that? By doing that, it changes the enzymatic activity of the protein. It also influences how these proteins bind to DNA, RNA, or other proteins. And it has been shown that by doing this kind of modification, PARP can promote DNA repair, it can maintain genomic stability by regulating chromatin structure, or it can even maintain telomere length. So as you know or may be aware of, the telomere length or deterioration of telomere length is a big leading cause of aging. So there's a connection between PARP activity and aging as well. For example, centenarians or the people who live over 100 actually show high ADP ribosylation capacity in their blood cells, which means their cells are actually repairing the DNA pretty quickly and pretty efficiently. Now the next prominent role of NAD is to regulate various metabolic reactions in different tissues. So some of these are its role in reducing neurodegeneration in the brain. Patients suffering from pellagra actually has dementia as one of their side effects. It is also cardioprotective. In liver, it helps in fatty oxidation and gluconeogenesis. It helps to burn your fat and reduces lipogenesis or formation of fat. In pancreas, it helps in insulin secretion. In muscle, it helps in mitochondrial activity. It improves insulin sensitivity. It also helps in fatty acid oxidation. And in my work, I actually have shown that NAD plays a key role in endothelial cell function as well. And by doing that, it actually reduces vascular aging phenotypes and also improves blood flow. And this was published in the journal Cell. And it also has beneficial effect on the kidney as well. So one of the way NAD can do this multitude of effects or can have multitudes of effects is by regulating the function of family of proteins called sirtuins. So sirtuins use NAD+ as a substrate. So just like PARP, it removes the acetyl modification and then removes that acetylricin residue and modulates the function of the protein. And in the process, it also releases nicotinamide, which shuttle back to NAD synthesis by salvage pathway. This reaction is called deacetylation reaction. And it was a big breakthrough in 2000 when Shinemai and Dr. Leonard Gorente found this reaction and linked NAD plus to sirtuin and showed that NAD plus can actually extend yeast lifespan. And my venture into this field of aging and NAD plus actually started in Professor Leonard Gorente's lab. As you can imagine, if your NAD plus level falls below the threshold level, what will happen? So you can have an increased DNA damage, you can have a redox imbalance, you can have increased insulin resistance, and it will also cause inflammation. Finally, you will develop all these diseases like cardiovascular diseases, neurodegenerative diseases, and musculoskeletal diseases. And this is exactly what happens with aging. So you can see here, unfortunately, NAD levels steadily decreases with age. In males, by the time you are 40, your NAD level is almost half of the amount as you were born. Same thing in female. And here in this work, I have shown that NAD levels decrease in mice, which is a model organism most frequently used in aging research. It not only decreases at the cellular level in endothelial cells shown here, but it also decreases in muscle, in gastroc muscle as well. The question is like, why does that happen? Why do NAD levels decrease with age? So one of the obvious reasons, of course, is the whole machinery that makes NAD becomes inefficient with aging. So the proteins get senile, just like we get senile, so it can't function properly, can't make NAD from the food sources like tryptophan, niacin, or NR. And then another reason that has been discovered recently is upregulation or increase in the activity of this protein called CD38. So CD38 is a NAD hydrolase. That means that it actually hydrolyzes NAD and destroys it. And its level goes up with aging. In fact, if you actually inhibit CD38 with a small molecule called epigenin, you can increase NAD plus level in your body and improve your metabolic function. So what are the other ways of increasing NAD? Because it's obvious from this point on that you need to restore the balance or keep the NAD plus balance in your body as you age to keep all the metabolic function running properly. So there are different ways of doing that. So one is changing your diet. So it has been shown that if you fast or restrict the calorie in your diet, you can increase NAD levels in your body. Calorie restriction should not be confused in malnutrition, because calorie restriction is getting enough nutrition but restricting the amount of calorie that you take. It's easier said than done, because you cannot be on calorie restricted diet throughout your life to increase NAD. Another good way of increasing NAD plus is exercise. So if you exercise, your mitochondrial activity goes up, your NAD plus is recycled very frequently, and you restore the homeostatic balance of different metabolic pathways. The third source, as we have talked about, is the food, of course. There are a lot of foods where niacin is found. The tuna fish is very rich in niacin. NR is obviously found in milk predominantly, and there are different vegetables and food where you can get NMN. But if you look at the number of NMN or the amount of NMN that's present in the food, you can understand that it's not enough to replace that declining NAD level that happens with aging. You need more of this. So one of the sensible way of doing that is actually to have these molecules as a dietary supplement. In fact, it has been found that if you give NR to mice, which is aging, it get increased amount of mitochondrial activity. So the mitochondria increases in size. The folds in mitochondria, which holds the electron transport proteins, actually increases with NR supplementation as well. That means that overall your mitochondrial activity is getting improved. NR has been tested in humans as well. The recent clinical trial, it was given to 70 to 80 years old human. It was well tolerated. And in fact, it reduced inflammation in the muscle. In my work, I have shown that if you give NMN as a supplementation through drinking water to old animals, 18-month-old mice, which is equivalent to 70-year-old human, the mice runs better on a treadmill for a longer distance, almost twice as much as a non-treated animal. It increases blood flow, it increases blood oxygenation, it also increases muscle vascularization, which is response to exercise. So starting from yeast until now, NAD has covered a big journey, has done a big journey. And it is an exciting phase of research right now. It's ever evolving. We are learning something about NAD every day. And if you really want to get involved in NAD and aging research, is a perfect time to be involved. There are a lot of human trials going on with molecules NMN and NR. I think all these results will be coming out soon. And one of the recent breakthrough in this field was identification of this protein, which is a mitochondrial NAD transporter, which solve the puzzle like how NAD plus is transported from cytoplasm into the mitochondria. So far, I've talked about the metabolism of NAD in our body, in the mammalian cells, and I've ignored the elephant in the room. So when we eat our food, it's just not us which breaks down the food and absorbs it and make NAD. There's another factor in our body that also utilizes the food and breaks it down and repurposes it. And it's a microbiome in our gut. So they also need NAD for their survival. They used a slightly different machinery to make NAD. And there's always a connection between the NAD synthesized by microbiome and the NAD synthesized by us. I cannot cover that in this lecture, but that will be for the next lecture. So I hope what I conveyed to you today was a brief history of nicotinamide or NAD plus research. I also told you how NAD plus is synthesized in our body. I showed you different ways that NAD is utilized, NAD plus is utilized in our body. I also told you how NAD plus or NAD plus levels decline with age and how you can actually restore it back, boost it by different ways. And finally, I told you about two molecules or two NAD precursors, supplementation of which actually improves some age-related function. Before I end my lecture, I want to leave you with a quiz, a very interesting quiz. So when pellagra was a problem in Europe, it was a problem in India as well, especially on the Deccan Plateau, where people actually ate jowar instead of maize. So unlike maize, jowar has a lot of tryptophan, but still people suffered from pellagra. Why? So I'd like to figure it out, but I'll give you one brief hint. It's a high level of leucine in jowar. And while solving this, you'll realize it's just not the nutrient itself, but the balance between them is also important, how it defines our health. I hope you enjoyed our lecture. I'll meet you next time again with more interesting stories on metabolism. Thank you for watching.